Hello everyone. In this section, I will introduce my study about the acute BHPF exposure and cardiac toxicity. Though BHPF is widely used in industrial manufacture, the effects of BHPF is known little to us. A paper reported that BHPF is anti-estrogenic and may cause adverse pregnancy outcomes in mice. However, the toxicity and effect on vertebrates is still not clear. As the introduction before, zebrafish is a commonly used model animal for acute toxicity experiments. Therefore, in this section, we try to learn the effect of acute BHPF exposure on zebrafish embryos and larvae. And this is the experiment flowchart. The fertilized eggs were collected and washed. Then the fertilized eggs were transferred into 12 well plates and exposed in different BHPF concentrations at 2 hours post-fertilization. The mortality was recorded and the morphology was observed at different developmental stages. The body length, cardiac development, and related gene expression was studied to learn the response to different concentrations of BHPF. The morphology of different developmental periods showed that acute BHPF exposure delayed embryonic development, and the high-level BHPF exposure led to death during the first 24 hours. At 10 hours post-fertilization, the epiboly would be completely in the end of the normal development stage. We take the epiboly rate as the indicator to evaluate the developmental state. The result showed that the completely epiboly rate, which was expressed as 100% epiboly, was obviously decreased at 10 hours post-fertilization in BHPF exposed group compared with the control group. In addition, the median length concentration was calculated at 1970 hours post-fertilization by probate analysis. Further observation found that acute BHPF exposure resulted in abnormal morphological phenotype. Just as the figure showed, BHPF exposure led to developmental malformation and caused precardial edema at 72 hours post-fertilization. The body length became obviously shorter than control group. Besides that, the tail curled when exposed under high levels of BHPF. At the same time, the head area and the eye area were also measured. The results displayed that BHPF treatment decreased the head area and the eye area. These results indicated that BHPF exposure damaged the development of zebrafish. Furthermore, the apoptosis was detected by acridine orange staining. The apoptosis points were concentrated in the regions of head, heart, and tail. The results showed that apoptosis was increased in head and tail under BHPF exposure in a concentration-dependent manner. Moreover, the cardiac morphology was further observed. The figure showed that acute BHPF exposure retargeted cardiac development and resulted in abnormal cardiac morphology. BHPF exposure increased precardial area and precardial edema rate. The distance of senior venous to bulbous arteriovenous, the distance of the SV to BA, one indicator of cardiac development, increased in a concentration-dependent manner. The transgenetic line special marketed the heart with GFP was used to directly show the morphology of heart. The increased SV to BA distance indicated the failure of cardiac looping. The ventricle and atrium look like in a line under high concentration of BHPF exposed, while in the control group, the ventricle and atrium looped and overlapped in the lateral review. The statistic results also indicated the failure of cardiac looping, and it was significant under the different levels of BHPF exposed. The malformed heart of BHPF exposed zebrafish was further confirmed by the histopathological examination by H.E. staining. Compared with the control group, obviously tubular heart was observed in a linearization of ventricle and atrium in BHPF exposed group. The cell number of the two campers also decreased under BHPF exposed. The effect of BHPF on cardiac function was further learned. The parameters of cardiac function, including heart rate, stalking volume, cardiac outcome of ventricle, and frictional shortening of ventricle, were reduced in BHPF exposed group compared to the control group. This indicated BHPF exposed can disturb the contractility of heart and affect cardiac function. The expression of cardiac development-related genes was further detected. To learn the mechanism of the BHPF on cardiac toxicity, the downregulation of cardiac development-relevant genes, including NTX2.5, TBX5A, GATA4, MYH6, and MYH7, indicated the mechanism of cardiac abnormal structure and function. This experiment showed the effect of acute BHPF exposed on early embryonic development of zebrafish. Results indicated that BHPF lead to developmental toxicity, induced abnormal embryonic developmental process, and increased cell apoptosis. Moreover, acute BHPF exposed induced cardiac toxicity, retarded cardiac development, led to failure of cardiac looping, and decreased the cardiac contractility, further affected the cardiac morphogenesis and function. As a result, BHPF exposed led to cardiac developmental malformation.